This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome to a captivating journey into the depths of our genetic past. For generations, the story of human origins seemed straightforward. But groundbreaking science now reveals a far richer, more complex truth. Our ancestors didn't just spread across the globe. They met, mingled, and interbred with other ancient human populations. This process, known as archaic introgression, left an indelible mark on our DNA. Join us as we uncover this incredible hybrid human story, exploring how these ancient encounters continue to shape our biology, health, and very essence today.
Chapter 1. Ancient Encounters, Our Hybrid Human Story. For a long time, the story of human origins was understood through a relatively simple lens. Anatomically modern humans, our direct ancestors, evolved exclusively in Africa around 300,000 years ago. From this African cradle, they embarked on a series of migrations, eventually spreading across the entire globe. This model, often called the out-of-Africa theory, suggested that as our ancestors ventured into new territories, they largely replaced any other archaic human populations they encountered, with little to no significant intermixing. It was a narrative of singular triumph, of one lineage moving forward. But science, as it often does, is constantly evolving, refining our understanding with new discoveries. In recent decades, particularly with the advent of advanced genomic sequencing technologies, this straightforward narrative has given way to a far richer, more complex, and utterly captivating saga. The story we now piece together is one of multiple migrations, ancient encounters, and intimate intermingling, a tapestry woven with threads from diverse human lineages, both modern and archaic. Our shared evolutionary history with other hominins, like Neanderthals and Denisovans, stretches back much further than we once thought, with a common ancestor living somewhere between 765,000 and 550,000 years ago. While our lineage was developing in Africa, these other archaic groups had already established themselves across vast stretches of Eurasia. The first significant waves of modern humans began to expand out of Africa by at least 85,000 years ago, though the major successful dispersal that led to widespread colonization of Eurasia occurred approximately 70,000 to 50,000 years ago. As these early modern human pioneers pushed into new continents, they didn't just replace the existing archaic inhabitants. They met them, interacted with them, and, crucially, they interbred. This process, known as archaic introgression, introduced genetic material from these ancient cousins into the modern human gene pool, leaving a lasting, indelible mark on our DNA, influencing everything from our immune systems to our adaptation to new climates. One of the most well-known archaic groups our ancestors encountered were the Neanderthals. These robust hominins had thrived across western and central Eurasia for hundreds of thousands of years, adapting to the diverse climates and landscapes of ancient Europe and the Middle East. Evidence from genomic sequencing, first published in 2010, dramatically confirmed what some paleoanthropologists had long speculated. Modern humans interbred with Neanderthals. These admixture events were not single occurrences, but happened episodically, likely in regions like the Near East, around 47,000 to 65,000 years ago, although some research suggests earlier contact up to 86,000 years ago. A powerful, concrete example of this direct interbreeding came with the discovery of Denisova 11, nicknamed Denny, a first-generation hybrid individual whose mother was Neanderthal and father was Denisovan. The genetic legacy of these encounters is still very much with us today. On average, most non-African populations carry just under 2% of their DNA from Neanderthals. However, the distribution isn't entirely uniform. Interestingly, studies have shown that East Asian populations tend to carry a slightly higher proportion of Neanderthal ancestry, sometimes estimated to be around 8% more than Europeans, referring to a relative increase in the proportion of Neanderthal ancestry. This intriguing difference has prompted scientists to consider various explanations, including the possibility of additional waves of interbreeding specifically with the ancestors of East Asians, or perhaps a dilution of Neanderthal ancestry in European populations due to later migrations of groups with less archaic DNA. Even within Africa, the genetic story is more nuanced than initially believed. While sub-Saharan African populations generally have very little Neanderthal DNA, trace amounts have been identified in North African populations and even some sub-Saharan groups. Specifically, African individuals have about 0.3% of their genome, or 17 megabases, composed of Neanderthal admixture, likely introduced through later back migrations of modern humans from Eurasia around 20,000 years ago. North African populations show varying levels, with Tunisian Berbers having 100 to 138% of the Neanderthal signal compared to Eurasian populations, North Morocco and Egypt around 60 to 70%, and South Morocco 20%. Low but significant rates of Neanderthal admixture have also been observed for the Maasai of East Africa.
But Neanderthals were not the only archaic relatives our ancestors mingled with. Deep in a Siberian cave, a tiny finger bone and a few teeth unearthed a completely new and enigmatic human group, the Denisovans. Much less is known about their physical appearance compared to Neanderthals, as fossil evidence is scarce, but their genetic story is equally compelling. Modern humans also interbred with Denisovans, with these admixture events estimated to have occurred around 44,000 to 54,000 years ago. The Denisovan genetic signature is generally less prevalent than Neanderthal DNA in most modern human populations, typically less than 1%. Mainland Asians and Native Americans, for instance, carry about 0.2% Denisovan ancestry, and South Asians have levels similar to East Asians, around 0.1%. However, there are striking exceptions. Some oceanic populations, particularly in Melanesia and among the Philippine Aita, carry a significantly higher proportion, sometimes up to nearly 5 or even 6% of their genome from Denisovan ancestors. This suggests a particularly rich history of interaction in these regions. Recent findings even indicate that Near-Oceanians show evidence of introgression from not just one, but 3 distinct Denisovan-like groups, painting a picture of diverse and repeated interbreeding events across Asia and Oceania. And the story doesn't end with just Neanderthals and Denisovans. The genomic landscape reveals even deeper mysteries, the presence of ghost populations. These are archaic human groups that scientists have inferred through statistical analyses of modern human genomes, leaving behind genetic echoes but no physical fossil record. These beings existed. They walked the earth and had children with our ancestors. For example, recent research suggests that some West African populations derive a significant portion, between 2 and 19%, of their genetic ancestry from an archaic population that diverged from our ancestors even before the split of modern humans and Neanderthals. These findings are truly mind-bending, suggesting that our ancestors encountered and interbred with unknown hominin groups that walked the earth, leaving their indelible mark on our DNA, even if their bones have yet to be discovered. Even the Denisovan genome itself holds secrets, containing about 4% of DNA from an unknown archaic human species that diverged over 1 million years ago. Furthermore, it's believed that the common ancestors of Neanderthals and Denisovans may have interbred with an even more ancient, unidentified, super-archaic human species, descendants of a very early migration out of Africa nearly 2 million years ago. These groundbreaking genetic discoveries have fundamentally reshaped the scientific consensus regarding human origins.
The strict out-of-Africa replacement model has been largely superseded by an assimilation model, which acknowledges that while the majority of modern human ancestry is African in origin, the gene pools of contemporary populations were significantly shaped by these ancient hybridization events. It's also clear that gene flow wasn't just a one-way street. Modern humans also contributed genetic material to archaic populations. For instance, approximately 6% of the genome of the Altai Neanderthal, an individual whose remains were found in Siberia, was inherited from modern humans around 250,000 years ago. The consequences of this extensive intermixing were far from simple. Some archaic DNA segments were found to be adaptively introgressed, meaning they conferred a fitness benefit to modern humans, helping them adapt to new environments outside of Africa. These beneficial genes saw rapid expansion within certain groups, providing ready-made genetic toolkits for challenges such as new pathogens, e.g. immunity genes like HLA alleles, OAS1, STAT2, TLRs, adapting to cold climates, e.g. keratin genes for skin and hair, TBX15/WARS2 for metabolism, or high-altitude environments, e.g. EPAS1 in Tibetans. This complex story of ancient encounters and genetic exchange is not merely a historical curiosity. It represents a profound shift in our understanding of what it means to be human, revealing that our lineage is a mosaic shaped by a deep and interconnected past. The genetic legacy of these archaic populations continues to influence our biology in ways we are only just beginning to uncover, from how our bodies function to our susceptibility to various health conditions, inviting us to explore the intricate details of this ancient inheritance in the chapters that follow.
Chapter 2. Ancient DNA, Shaping Our Intimate Biology. Building on our exploration of ancient encounters and the pervasive presence of archaic DNA, this chapter delves into its profound and often surprising influence on the most fundamental and intimate aspects of our biology. From the very processes that create new life, to the rhythms that govern our daily existence, and even the architecture of our brains, ancient genes are still actively shaping who we are. Let's start with something truly fundamental: human reproduction and development. While some archaic DNA was selectively pruned from our genome, a more complex and often beneficial integration of archaic DNA into many reproductive processes also occurred. Imagine for a moment the incredible complexity of creating a new human life. It involves intricate choreography at the genetic level, from the development of sperm and egg through the delicate stages of embryogenesis and into fetal development. Scientists have now identified 118 genes within 47 archaic segments, collectively spanning nearly 38 million base pairs, 37.88 megabits, of our genetic code that are associated with reproduction, either in humans or in other mammals like mice, and show clear evidence of adaptive introgression from archaic humans. This isn't just a handful of random genes. These are segments of DNA that were not only passed down, but actively favored by natural selection because they conferred some fitness benefit. Furthermore, this genetic legacy isn't uniformly distributed across the globe. Researchers have observed regional specific patterns, with 26 segments in American, 17 in East Asian, 6 in European, 1 in Middle Eastern, and 6 in Oceanic populations.
Among these, researchers have pinpointed 11 archaic core haplotypes, groups of genes inherited together from our ancient relatives, with 3 of them showing strong signs of positive selection. What's truly astonishing is that hundreds of archaic alleles within these regions have a genome-wide significance for a wide array of traits. Even more compelling, over 300 of these archaic variants act as EQTLs, or expression quantitative trait loci. This means they directly influence the activity levels of 176 different genes. And here's the kicker. A remarkable 81% of these archaic EQTLs located within these core haplotypes specifically regulate genes that are expressed in reproductive tissues, such as the ovaries, prostate, testes, and vagina. This is a powerful indication that ancient DNA is not just passively present, but is actively involved in controlling the very machinery of human reproduction. Further research highlights that some of these introgressed genes are also enriched in crucial developmental pathways, influencing how our bodies are built and function throughout our lives. For instance, while some archaic genes might be linked to reproductive challenges like preeclampsia, which we'll explore in Chapter 4, others offer protection. This suggests a complex legacy where archaic genes can both introduce vulnerabilities and offer unexpected resilience in our most fundamental biological processes. Beyond reproduction, archaic DNA has even woven itself into the very fabric of our cellular machinery in ways that were previously overlooked. You might be familiar with mitochondrial DNA, the genetic material found in the powerhouses of our cells, inherited exclusively from our mothers. But did you know that fragments of this mitochondrial DNA can sometimes integrate themselves into our main nuclear genome? These are called nuclear mitochondrial DNA segments, or NUMTs. For a long time, these NUMTs were considered largely inert, genomic fossils, interesting relics but not active players in our biology. However, recent studies have completely reframed this understanding. Scientists have systematically characterized hundreds of these polymorphic NUMTs across diverse human populations, and crucially, they've identified 5 specific NUMTs that were introduced into modern humans directly through archaic introgression. These aren't just passive pieces of ancient code. These integressed NUMTs can profoundly alter how our nuclear genome functions. They can modulate gene expression, for example, leading to the upregulation of an immune-related gene called RASGRP3. They can also reshape the three-dimensional structure of our chromatin, the tightly packed DNA within our cell nuclei at important locations, influencing genes like SCD5 and HNRNPD. This changes our view of NUMTs from mere genomic footnotes to dynamic elements actively influencing human diversity and adaptation, a truly intimate integration of archaic fragments into our core genetic operating system. And the influence extends to the rhythms of our daily lives. Think about your internal body clock, your chronotype. Are you a morning lark or a night owl? This preference, along with our sleep-wake cycles, is governed by our circadian rhythm, a fundamental biological process that cycles roughly every 24 hours. Modern humans and our archaic relatives, like Neanderthals and Denisovans, evolved separately for hundreds of thousands of years in vastly different environments, including varying latitudes with distinct light-dark cycles and seasonal changes. This led to divergent adaptations in their circadian biology. Scientists have now traced the evolution of chronotype by comparing archaic and modern human genomes. They've identified dozens of circadian genes that contain variants with the potential to alter gene splicing or regulation in archaic hominins, including key genes like clock, PER2, RORB, RORC, and ROA. This suggests that archaic introgression could indeed modify circadian gene expression in modern humans, and the evidence is striking. Introgressed variants are significantly enriched among the genetic switches that regulate circadian genes. The strongest effects of these introgressed archaic alleles on our chronotype tend to increase morningness. This is a fascinating finding, consistent with adaptations to higher latitudes, where daylight hours fluctuate dramatically with the seasons. These variants are also associated with affective disorders and are significantly enriched in immunity pathways, further supporting adaptations to strong seasonal light variations. It suggests that as our ancestors migrated out of Africa and into these new environments, archaic genes provided a valuable adaptive edge, perhaps by fine-tuning our internal clocks to better cope with extreme seasonal changes in light availability. Some of these archaic variants even appear to enhance serotonin synthesis, a crucial chemical for regulating mood and sleep. So, that feeling of being a morning person might just be a whisper from your ancient Neanderthal or Denisovan ancestors. But not all archaic legacies are beneficial. Sometimes these ancient genetic contributions can predispose us to certain health challenges or influence our fundamental anatomy in unexpected ways. Take, for instance, the complex architecture of the human brain and skull. Headaches, dizziness, balance problems, and blurred vision are common symptoms of Chiari malformations, structural defects where the lower part of the brain extends into the spinal cord. Intriguingly, research comparing modern human skulls with those of our ancient relatives has revealed a striking connection. Individuals with Chiari type 1 malformation often have skulls with a smaller, flatter occipital bone at the back and base, leading to increased brain compression, a skull shape that closely matches that of Neanderthals. This archaic Homo introgression hypothesis suggests that some of the genes inherited from Neanderthals, particularly those involved in cranial development, could be contributing to these modern brain malformations. This is a powerful reminder that the intermingling of ancient lineages can have lasting and sometimes detrimental impacts on our most vital organs. Beyond specific malformations, Neanderthal admixture has also been associated with subtle changes in our brain morphology, influencing areas like the visual cortex and regions involved in visual processing. Moreover, studies comparing gene expression have found that archaic haplotypes tend to have lower expression levels in the brain, hinting at deeper genetic incompatibilities that likely shaped the early survival of these intergressed segments. Even our metabolism, the very way our bodies process nutrients, carries the fingerprints of archaic introgression. Consider the essential micronutrient vitamin B1, or thiamine. Genes involved in thiamine metabolism are crucial for cell function, energy production, and immune response. In East Asian populations, scientists have found strong signals of positive selection in several genes related to thiamine processing, specifically SLC19A2, SLC35F3, and SLC35F4. The adaptive haplotype in SLC35F4, for example, is highly likely to be of Neanderthal origin, while another in SLC35F3 also traces back to an archaic ancestor. These beneficial archaic variants became highly frequent over 10,000 years ago and persist in modern East Asian populations, suggesting they provided a crucial advantage. Initially, this adaptation was likely driven by challenges like pathogens rather than the later development of agriculture, highlighting how ancient genes helped our ancestors survive in new environments. Other metabolic adaptations include variants related to lipid metabolism and body fat distribution, such as a Denisovan variant of the TBX15 gene, found at high frequency in Greenlandic Inuit, which may have helped them adapt to cold temperatures by influencing how their bodies store and use fat. From the intricate dance of reproduction to the subtle daily rhythms of our bodies, and even the very structure of our brains and our metabolic efficiency, archaic introgression is a pervasive force. It's a testament to the deep, enduring genetic legacy of our ancient relatives, constantly shaping our biology in ways we are only just beginning to fully comprehend. As we continue to unravel these ancient genetic threads, what further insights will they offer into the intricate origins of human health, traits, and our very essence today? Chapter 3. The Ancient Blueprint of Reproduction. Building on our previous chapter's exploration of archaic introgression and its broad influences, we now narrow our focus to understand how these ancient genetic contributions are dynamically shaping the function of modern human reproductive organs. For a long time, the prevailing narrative suggested that much of the archaic DNA related to reproduction was purged from our genome, particularly from the X chromosome and genes expressed in the testes. This was thought to be evidence of hybrid incompatibility, leading to reduced fertility in early admixed individuals.
While that selective pruning certainly occurred, and we'll delve into its implications shortly, recent scientific investigations have unveiled a more complex and frankly astonishing truth. A substantial portion of archaic DNA has not only survived in genes associated with reproduction, but has been actively incorporated and even favored, playing a crucial role in the very machinery that creates new life. As explored in Chapter 2, scientists have identified numerous archaic genetic segments in modern humans that overlap with genes involved in reproduction, with some archaic variants occurring at dramatically higher frequencies in certain populations, suggesting a strong adaptive advantage. This genetic legacy isn't uniformly distributed, showing regional specific patterns across the globe. Researchers have pinpointed archaic core haplotypes, some showing strong positive selection, and many archaic alleles were genome-wide significant for a variety of traits, with a substantial number of these variants acting as EQTLs regulating other genes.
The real revelation, however, lies in understanding the mechanism of this active shaping. As discussed in Chapter 2, archaic EQTLs, expression quantitative trait loci, act as genetic switches, regulating the activity levels of genes. What's truly remarkable is that many of these archaic EQTLs found within advantageous genetic bundles inherited from our ancient relatives specifically regulate genes expressed in our reproductive tissues. Imagine the intricate dance of cells and hormones within the ovaries, orchestrating the development of eggs and the delicate balance of female hormones. Or picture the testes tirelessly producing sperm and testosterone, crucial for male fertility. And consider the prostate and vagina, organs vital to reproductive health and function. When archaic EQTLs actively regulate genes in these specific tissues, they are directly influencing the fundamental biological processes that underpin reproduction. This could mean fine-tuning the timing of ovulation, altering the quantity or quality of sperm, modulating hormone production, or even influencing the cellular environment necessary for successful fertilization and early embryo development. This isn't just a historical echo, it's a living, breathing genetic instruction set, inherited from our archaic cousins, that is actively contributing to the daily function and overall health of our reproductive organs.
Beyond the immediate mechanics of reproduction, these adaptively introgressed genes are also deeply embedded in critical development pathways. Several of these adaptively introgressed genes are enriched in developmental and cancer pathways, and some have been associated with embryo development. This means their influence isn't limited to a single event like fertilization, but extends throughout an individual's entire lifespan. From the initial formation and maturation of reproductive organs during development, through the onset of puberty, and into the complexities of reproductive aging, these archaic alleles appear to play a regulatory role. They contribute to the intricate genetic programs that ensure these vital organs develop correctly, maintain their function, and respond appropriately to physiological changes over decades. This suggests a continuous, dynamic interaction between archaic and modern human DNA, perpetually optimizing and adapting our reproductive systems. But the story of archaic DNA shaping our reproductive organs isn't solely about beneficial additions. It's also about what was purged and why, a complementary form of evolutionary sculpting that refined our reproductive biology. While some models suggest that widespread reproductive incompatibilities between modern humans and archaics may not have occurred, strong evidence still points to specific challenges, particularly for male fertility. This phenomenon, often termed hybrid incompatibility in this context, led to strong negative selection against certain archaic genes. The evidence for this is stark. Large regions of our X chromosome, for instance, show a significantly reduced amount of Neanderthal ancestry compared to other chromosomes, with on average 7 times more archaic coverage on autosomes than chromosome X. This depletion also shows broad continental patterns, being greatest in European samples and least in South Asian samples. Furthermore, there was an excess of modern human DNA in the X chromosomes of Neanderthals and very little Neanderthal inheritance in the X chromosome of modern sapiens, providing a direct genetic mechanism for this observed bias. The X chromosome, with its unique inheritance pattern, is particularly vulnerable to such incompatibilities. In males who carry only one X chromosome, recessive deleterious archaic variants on this chromosome would have been directly exposed to selection, leading to their efficient removal if they compromised fitness. Furthermore, genes that are highly expressed in the testes, organs central to male reproduction, also show a marked depletion of archaic DNA. This pattern strongly suggests that many archaic genetic variants, while perhaps benign or even beneficial in their original archaic genetic background, were detrimental when combined with modern human genes, leading to reduced male fertility in hybrids. This active removal process wasn't a passive loss. It was a powerful evolutionary force that shaped the functional genetic landscape of our reproductive organs, weeding out incompatible elements and ensuring the viability of future generations. Ultimately, the genetic legacy of archaic introgression within our reproductive organs is a testament to evolution's pragmatic nature. It's a story of both gain and loss, of adaptation and refinement. Our reproductive systems today are not solely the product of our own lineage's evolution, but a finely tuned biological apparatus that has been significantly influenced, enhanced, and sculpted by the genetic contributions of our ancient cousins. These ancient genes continue to actively regulate fundamental processes, from the cellular level to the overall development and health of our reproductive organs throughout our lives. What other hidden influences might these ancient genes hold, waiting to be discovered, as we continue to unravel the deeper story of our reproductive biology? Chapter 4. Reproduction's Ancient Paradox. Building on our understanding of archaic introgression, this chapter delves into a profound paradox: how this ancient genetic legacy acts as a double-edged sword, providing evolutionary advantages while simultaneously predisposing modern humans to challenging health conditions, particularly reproductive disorders. Imagine a genetic inheritance that offers both a shield and a vulnerability. This is precisely what we find when we delve into the archaic contributions to human reproductive health. Let's start with some of the more challenging aspects, conditions that affect millions of individuals today. Recent scientific endeavors have unearthed compelling evidence linking archaic introgression to reproductive inhibiting phenotypes, such as endometriosis and preeclampsia. These aren't just casual associations, they are deeply rooted in the genetic architecture we inherited from our ancient cousins. Endometriosis, a chronic and often debilitating condition where tissue similar to the lining of the uterus grows outside the uterus, can cause severe pain, infertility, and other complications. Preeclampsia, a serious pregnancy complication characterized by high blood pressure and potential organ damage, poses significant risks to both mother and baby. The very genes that were adaptively introgressed, meaning they were positively selected for their beneficial effects, have, in some contexts, also been associated with these reproductive disorders. This highlights a crucial point in evolution. What might have been advantageous in one environmental or genetic background can become detrimental in another, especially over tens of thousands of years of human evolution and changing lifestyles. The same ancient genetic switches, or EQTLs, that regulate genes expressed in reproductive tissues can, under different circumstances, contribute to the complex genetic predispositions for these challenging conditions. Consider another fascinating and stark example of this double-edged nature within the context of pregnancy. A specific Neanderthal variant found in the PGR gene, which plays a role in progesterone signaling, was initially linked to an increased risk of preterm birth in some European populations. On the surface, this might seem like a clear disadvantage, a harmful genetic legacy. However, a deeper, more comprehensive analysis of the entire Neanderthal haplotype within this PGR gene revealed a remarkable twist.
This same archaic haplotype was actually associated with a reduction in the number of miscarriages and a decrease in bleeding during pregnancy. So, while a single variant might raise concerns, the broader genetic package from Neanderthals could in fact enhance fertility and pregnancy success within a modern human genetic background. It's a powerful illustration of how the effects of archaic genes are not always straightforward, but often depend on the precise genetic context and the specific combination of alleles. The complexities continue with other archaic genetic contributions impacting childbirth. Scientists have identified a specific Neanderthal allele, SNP 3,917,862 rupees, that is associated with hypercoagulability, a tendency for blood to clot excessively. This condition can be harmful, leading to serious health issues like strokes or deep vein thrombosis. Yet, the very same archaic allele offers a surprising protective effect. Women who lack this allele are a small but significant fraction more likely to die in childbirth. This is a stark example of a single ancient gene carrying both a potential health risk and a life-saving advantage, particularly in a critical reproductive event like childbirth. It forces us to confront the intricate trade-offs that natural selection has navigated over millennia, where a gene might be maintained in the population because its benefits, even if subtle, outweigh its potential drawbacks in certain crucial scenarios. These examples underscore the ongoing evolutionary tug-of-war that has shaped our genomes. While certain archaic genes undoubtedly conferred fitness benefits, allowing our ancestors to thrive in new environments, the process of introgression also introduced genetic material that was not perfectly compatible with the modern human genetic background. This led to a continuous process of selective pruning, where many archaic alleles were purged from our genome, particularly those that caused specific genetic challenges. Beyond specific diseases, archaic genes have also influenced fundamental aspects of our reproductive lifespan. Research has shown that introgressed enrions contribute to the heritability of several complex traits, including the age of menopause. While the precise nature of this influence, whether it tends to accelerate or delay menopause, can be complex and population-specific, it highlights how archaic genes can modulate a critical reproductive milestone. In different environments and social structures, an altered age of menopause could have offered either an advantage by extending the reproductive window, or a disadvantage, depending on the survival and child-rearing strategies of ancient populations. Ultimately, the story of archaic introgression is one of profound evolutionary trade-offs. Our ancient cousins, through their genetic contributions, have equipped us with a remarkable toolkit for survival and adaptation, but they have also bequeathed us certain predispositions that manifest as health challenges today. Understanding this delicate, double-edged balance is not merely an academic exercise. It's a vital step in unraveling the intricate origins of human health and disease. As we continue to delve deeper into our archaic genetic mosaic, what further complexities and unexpected connections will we uncover between our ancient past and our present-day biology? Chapter 5: Ancient Genes: Shaping Life, Fighting Cancer. Welcome back, everyone. Our journey through the genetic echoes of the past has so far revealed how archaic DNA intricately shapes our reproductive processes, our daily rhythms, and even the fundamental architecture of our brains. But the pervasive influence of ancient introgression extends even further, reaching into the very blueprints of human development, and, surprisingly, affecting our susceptibility to serious diseases like cancer. This chapter will uncover how these inherited genetic fragments play unexpected and sometimes contradictory roles in building our bodies and defending them against illness throughout our lives. It's a testament to the profound and often surprising impact of our ancient genetic heritage on our health today. Let's begin by exploring how archaic introgression has woven itself into the fabric of human development.
Beyond the immediate processes of reproduction, these ancient genes appear to regulate crucial developmental pathways that guide the formation and function of our bodies from conception through adulthood. For example, research has highlighted that several genes identified as adaptively introgressed from archaic hominins are significantly enriched in pathways critical for overall development. This suggests that the evolutionary benefits conferred by these ancient genes were not limited to specific traits, but contributed to the fundamental processes that ensure healthy growth and maturation.
A striking illustration of this developmental impact can be seen in our skeletal structure. A specific gene called TRPS1, which is involved in skeletal development, shows clear evidence of adaptive introgression from Denisovans in populations from near Oceania. This same gene also exhibits signs of strong positive selection in other geographically diverse groups, including rainforest hunter-gatherers in Central Africa and highland populations in Ecuador. This suggests that this Denisovan-derived variant provided an adaptive advantage related to bone formation and growth, potentially aiding our ancestors in adapting to varied environmental pressures or dietary needs across the globe. Similarly, archaic DNA influences aspects of brain development beyond the structural malformations we touched upon in a previous chapter. Studies of Denisovan genetic contributions in Papuan populations living in the highlands, for instance, have identified variants that are linked to early brain development. This indicates that these ancient genetic contributions may have played a role in shaping the neurological foundations laid during the earliest stages of human life, potentially influencing cognitive capacities or brain architecture in ways unique to these populations.
Furthermore, the archaic influence on our circadian rhythms, our internal biological clocks, extends to our long-term health and developmental trajectory. While the effects on morningness and adaptation to high latitudes were previously discussed, the implications for lifespan development are equally significant. A misalignment of our circadian clock, whether due to genetic predispositions or modern lifestyle factors, has been linked to an increased risk of developing a range of chronic health issues, including obesity, diabetes, and notably certain types of cancer. This means that archaic variants that helped fine-tune our ancestors' internal timing mechanisms might also, in the context of modern life, influence our vulnerability to these conditions that manifest over years, shaping the developmental landscape of our health across an entire lifetime. Now, let's turn our attention to one of the most critical aspects of human health, susceptibility to cancer. The role of archaic introgression here is surprisingly multifaceted, offering both protective benefits and complex predispositions. One of the most remarkable findings is the direct protective effect of certain archaic alleles against prostate cancer. Research has specifically identified archaic alleles overlapping an introgressed segment on chromosome 2 that act as a safeguard, reducing the risk of this common male cancer. This is a powerful example of ancient DNA providing a tangible, beneficial impact on modern human health, actively contributing to our resilience against disease. It underscores the idea that not all archaic genetic contributions were detrimental or neutral. Some offered clear advantages that natural selection favored and preserved. Beyond this specific protection, a broader picture emerges. Several of the adaptively introgressed genes found in modern humans are significantly enriched in developmental and cancer pathways. This doesn't necessarily mean they cause cancer, but rather that these archaic genes are intimately involved in the complex regulatory networks that govern cell growth, differentiation, and programmed cell death, processes that, when dysregulated, can lead to cancer. Their presence in these pathways suggests a deep evolutionary history where archaic variants may have been co-opted or maintained due to their beneficial roles in development, even if this involvement carries a nuanced relationship with cancer risk in modern contexts. It points to a delicate balance where the same genetic elements that ensure proper development might also, under certain conditions, contribute to the intricate mechanisms of disease. Adding another layer to this intricate relationship, the evolutionary history of oncogenic viruses, those capable of causing cancer, is also intertwined with archaic introgression. For instance, studies have revealed a transmission of oncogenic human papillomavirus 16, or HPV-16, between archaic and modern human ancestors. This finding suggests that archaic introgression may have influenced not just our innate genetic makeup, but also the coevolutionary dynamics between humans and pathogens, including those with cancer-causing potential. Understanding this ancient interplay can provide insights into the current prevalence and genetic diversity of such viruses in human populations. Furthermore, the indirect connections between archaic introgression and cancer are equally compelling. Our immune system, a crucial line of defense against disease, including cancer, has been profoundly shaped by archaic DNA. Many introgressed variants, particularly those acting as EQTLs, are significantly enriched in immunity pathways. A robust and well-regulated immune response is vital for detecting and eliminating cancerous cells before they can proliferate. The archaic contributions to our immune function, discussed in earlier chapters, thus play an indirect but critical role in cancer surveillance and prevention. Even our metabolic efficiency, previously highlighted for its role in adaptation, as discussed in Chapter 2, particularly the Neanderthal-derived genes that improved thiamine, or vitamin B1, metabolism in East Asian populations, has indirect implications for cancer susceptibility. Thiamine is an essential micronutrient vital for cell functioning and energy metabolism. Deficiencies can contribute to various health problems. By enhancing thiamine processing, these archaic genes improved overall cellular health and energy production, factors that are fundamental to preventing cellular damage and dysregulation that can lead to cancer. This illustrates how archaic adaptations, initially beneficial for survival in challenging environments, continue to support foundational biological processes that contribute to our long-term health and disease resistance. In essence, the legacy of archaic introgression is a testament to the profound and complex ways our ancient past continues to shape our present and future health. These archaic genes are not mere passengers, they are active participants, influencing the intricate dance of human development from the very first cells, and dynamically modulating our susceptibility and resilience to diseases like cancer throughout our lives. What further secrets do these ancient genetic fragments hold, waiting to be unlocked, that could provide even deeper insights into the origins of human health and disease? Chapter 6, Our Evolving Genetic Tapestry. Welcome to our final chapter, a culmination of our deep dive into the fascinating world of archaic introgression. We've journeyed through the ancient past, exploring how our modern human ancestors encountered and interbred with Neanderthals, Denisovans, and even mysterious ghost populations. We've seen how this ancient DNA has profoundly shaped our most intimate biological processes, from the very blueprint of reproduction to the subtle rhythms of our daily lives and even our susceptibility to disease. Now it's time to synthesize these incredible discoveries and understand how they are fundamentally rewriting the story of human evolution, not as a simple linear progression, but as a rich, complex tapestry woven with both invaluable advantages and unexpected vulnerabilities. As established in Chapter 1, the prevailing narrative of human origins has been fundamentally rewritten, from one of simple replacement to a complex story of assimilation and profound genetic intermingling. This shift means acknowledging that our survival and success were not achieved in isolation, but through a dynamic interplay with our ancient cousins, whose genetic contributions became an integral part of who we are. Archaic introgression offered a rapid shortcut for adaptation, providing our ancestors with ready-made genetic toolkits from Neanderthals and Denisovans already adapted to Eurasian environments, a mechanism that allowed for a much faster rate of adaptation than de novo mutations alone. This enabled our ancestors to thrive in diverse and challenging landscapes. As detailed in previous chapters, these adaptive advantages span crucial biological functions. They include enhanced immune responses and fine-tuned skin and hair characteristics. Archaic contributions also improved metabolic pathways. Even our circadian rhythms were recalibrated by archaic variants. These fundamental adaptations facilitated our global expansion, underscoring the immense evolutionary value of tapping into a broader shared gene pool. However, the rewritten story also comes with a sobering counterpoint: the evolutionary costs and trade-offs of this archaic inheritance. While introgression offered benefits, it was far from a perfect exchange. Archaic populations, with an effective population size about tenfold smaller than that of modern humans, often carried a greater deleterious load, an accumulation of slightly harmful genetic variants that natural selection was less efficient at removing. When these variants were introduced into the larger, more genetically diverse modern human populations, they became targets for purifying selection, actively pruning many archaic segments from our genome, especially those that proved incompatible with the modern human genetic background. This legacy, however, was a double-edged sword. As explored in chapters 2 and 3, archaic populations carried a greater deleterious load, leading to introgression deserts on the X chromosome and in genes highly expressed in the testes, suggesting reduced male hybrid fertility due to epistatic incompatibilities, as predicted by the Dobzhansky-Mueller model of speciation. Yet, this dynamic process of genetic refinement also saw archaic genes offering protection, while others introduced vulnerabilities, contributing to conditions like Chiari malformations or reproductive disorders such as endometriosis and preeclampsia, as detailed in chapters 2 and 4. It's a constant reminder that evolutionary solutions are often compromises, with benefits and drawbacks intertwined within the same genetic threads. The rewriting of human evolution is also deeply intertwined with our continuously advancing scientific methodologies. It's not just what we're discovering, but how we're discovering it. New computational tools and analytical frameworks are allowing researchers to peer deeper into our genomes, distinguishing between subtle signals of introgression and shared ancestral variation. Methods like ancestral recombination graphs, e.g. TRACE, and specialized algorithms that can identify archaic segments without needing a direct archaic reference genome, e.g. DISEG, are refining our maps of ancient ancestry. These innovations are enabling us to trace multiple waves of introgression, pinpointing when and where these ancient exchanges occurred with unprecedented precision. They are revealing that the genetic landscape of archaic contributions is far more dynamic than a static snapshot, constantly being shaped by ongoing selection, drift, and demographic events. This ongoing scientific quest is far from over. Each new high-coverage archaic genome, each new data set from previously underrepresented modern human populations adds another layer of detail to our complex evolutionary story. The study of archaic introgression is not just about looking backward, it's about understanding our present and future. By unraveling the beneficial and detrimental legacies embedded in our DNA, we gain critical insights into the genetic basis of human health and disease today. This deeper understanding can inform medical research, guiding us toward better diagnostics and treatments by recognizing the ancient roots of modern conditions. Ultimately, the story of human evolution, as rewritten by archaic introgression, is a testament to the power of intermingling, adaptation, and continuous change. It's a narrative that celebrates the incredible resilience and adaptability of our species, not despite our mixed ancestry, but often precisely because of it. We are not just descendants of out-of-Africa pioneers. We are the living, breathing embodiment of a vast, interconnected, and still unfolding saga of ancient encounters that continue to shape our biology, our health, and our very place in the world. The echoes of these ancient humans resonate within us, a vibrant, complex legacy that challenges us to look beyond simplistic narratives and embrace the extraordinary richness of our shared evolutionary journey. We've journeyed through the incredible story of archaic introgression, revealing how our ancestors' encounters with Neanderthals, Denisovans, and even unknown hominins fundamentally rewrote human evolution. This ancient genetic exchange wasn't a simple replacement, but a complex assimilation, providing vital adaptive advantages for immunity, metabolism, and climate, while also leaving us with intriguing predispositions to certain health conditions. Our biology is a rich mosaic, a living testament to this shared past. Understanding this intricate legacy isn't just about history. It's about unlocking deeper insights into our health and identity today. Thank you for joining us on this exploration of our evolving genetic tapestry.